My name is Daryl Hondorp. I'm a fishery research biologist with the USGS Great Lakes Science Center out of the Ann Arbor office. Big thank you to Susan and the committee for the invitation to talk to you this morning. Before I get started, just like to thank the following individuals and organizations for their support with some of the projects that the results we'll be sharing this morning. These are huge multi-collaborative efforts that just wouldn't be possible without help from a lot of people. So a big thank you to everybody listed here. So by now it's probably shouldn't be much of a surprise to you that lake whitefish across Lake Michigan and Lake Huron are in trouble. Both harvest and population sizes have declined precipitously. We're pretty sure that this is related to poor recruitment or failed recruitment, but mechanisms and the particular life stages affected are still. Is this not on? Yeah.
There we go.
All right, testing. All right, that sounds better. All right. Just to review, whitefish not doing well. The particular life stages and mechanisms involved in those recruitment failures are still being worked out, but managers are still tasked with wisely managing these populations and protecting them during this peculiar phase in their history. Mark Ebner and his colleagues in a 2021 white paper made a number of recommendations or highlighted some information needs that would be useful to managers trying to protect these fisheries in this particular time. One of the things they noted it would be good to have better information on stock structure of all the lake whitefish stocks in these two lakes. The logic here being, if stocks are dwindling, then the last thing you want to do is, set some kind of harvest limit that might further make them vulnerable to overfishing. Secondly, they called for exploring options for protecting and restoring spawning habitat. Obviously, anything you can do in that front to either prevent recruitment from sliding further or maybe enhance it could be a positive thing. The problem here is that our understanding of whitefish movement and habitat use is not that great at the moment. What we do know, based on tag return type studies that have a number of issues. Chief among these are low tag return rates, lack of repeat observations in the same individual, and also distributions and movement information that's biased by the distribution of fishing effort, because most of the tag returns come from commercial or recreational harvests. So this morning I'd like to share some results from a couple of projects that are using acoustic telemetry to track movements of lake whitefish. Chris Vandergoot did a nice job of giving an overview of this technology yesterday afternoon, but in case you weren't here, in acoustic telemetry, fish are captured. They're surgically implanted with an electronic transmitter or tagged, and then released back into the environment, where their movements are tracked on networks of autonomous hydrophones, often called receivers. And the nice thing about acoustic tags relative to, say, a pit or radio tag is they're very high powered, so they can be detected from greater distances, which gives you a lot more power to quantify movements at a variety of spatial scales. So the first project I'd like to share some results from is a project that's focused on looking at stock structure of lake whitefish stocks in northeast Lake Michigan, particularly those that spawn in Grand Traverse Bay and in adjacent areas of the northern Maine Basin. These probably aren't the largest stocks out there, but current genetic research suggests that there's a lot of genetic diversity in this particular group, so they're important for that reason. This project has a number of objectives, but the one I'd like to discuss this morning is just looking at telemetry, how telemetry can help validate whether movements of these fish correspond with the particular units of management consideration that we've drawn around them. And so This graph here shows two management zones, whitefish management zone 5, which is in the pink there, and then kind of the northeast portion of whitefish management zone 6, which is that blue color. Whitefish management zone 5 is kind of a curious spatial beast in that it includes all of Grand Traverse Bay, but also a huge chunk of water outside of the bay and the main basin. One of our main interests here are to ask to determine the extent to which these bay spawning fish intermix with these spawners in whitefish management zone 6, and also because of some previous work to look at how the degree to which these groups on the east and west sides, east and west shores inside Grand Traverse Bay mix together. To do that, we've acoustically tagged fish at 3 known spawning locations in the system. One is the Good Harbor Bay Reef, which is that well, it's probably hard for you to see because it's a little washed out up here, but there's a yellow diamond in that blue area. We sampled fish there, also in tagged fish there, also at 2 spots inside the bay, Ingalls Point, which is on the west shore, and Elk Rapids, which is on the east shore. All these fish were captured during the fall spawning season. The results that follow are based on 57 individuals that were tagged and released at Good Harbor Bay in the fall of 2021, 82 individuals that were tagged and released at Ingalls Point in 2021, and then a smaller sample of fish that were tagged and released at Elk Rapids in 2022. And then these X's show the distribution of acoustic receivers across these 2 zones. The nearshore receivers and the receivers inside Grand Traverse Bay were deployed by those of us who were working on lake whitefish projects, and then the offshore receivers and those in the island archipelagos were deployed by a group studying lake trout use of the Northern Refuge, led by Chuck Bronte. So this is also a nice example of 2 projects working together to extend receiver coverage beyond what either project could do by itself. So let's jump into some results here. This shows the distributions of acoustic tagged whitefish from the Good Harbor Bay population across the top and the Ingalls Point population across the bottom for select periods during 2022. There's a few points I want to make here is that firstly, fish that were tagged and released inside of Whitefish Management Zone 5 and 6 in general tended to remain within those zones, as you would hope would happen based on how these zones are drawn. Secondly, the highest concentrations of fish, again, which are probably a little hard to see because the figure is small, are really located very near the spawning sites, which suggests these fish are showing quite a bit of fidelity to the spawning site even during non-spawning periods. Thirdly, the greatest dispersal from the spawning site tends to occur during the summer, and that's also when there's the greatest overlap of fish between these 2 zones, but that tends to occur only in a very restricted area along the west coast of the Leelanau Peninsula there in the northeast corner of Whitefish Management Zone 6, but this didn't involve that many individuals. Lastly, there's not a lot of dispersal of these Ingalls Point fish out of the bay into the main basin portion of Whitefish Management Zone 5. Now jumping in to look at just those populations that spawn inside the bay, and this is data for 2022, just the winter and spring. Here we see that these fish on the west and east coast tend to kind of remain on their side of the bay. There's a little bit more dispersal from those west shore fish over in the east side, but these Elk Rapids fish are very restricted to the eastern shore, and they were rarely, if ever, detected on receivers on the west side of Old Mission Peninsula there. And again, neither group seems to disperse much into that main basin portion of Management Zone 5. So some of the things we've got going on here that we can say so far is that these fish tend to, at least for the moment, the data we have seems to suggest that their movements validate the unit stock assumption of statistical catch at age models. They show quite a bit of fidelity to their spawning sites, suggesting that they probably also show high spawning site fidelity, which is something we're going to quantitatively measure moving forward. There is a bit of a question as to whether these bay fish, maybe the movements almost suggest that maybe the bay could be its own management zone given the limited amount of migration out of the bay by those Elk Rapids and Ingalls Point fish. And lastly, these movements tend to reinforce some of the genetic differences that previous folks have seen amongst these populations. Justin Vandehey in 2009 released a paper where he found that these, based on analysis of neutral genetic markers, that these Elk Rapids fish were very distinct, and he went as far as to suggest that perhaps they should be its own genetic management zone. And then more recently, folks in Jared Homola and Wes Larson's lab have used genomics on the same group of fish, and here they found that the Ingalls Point fish were really unique genetic outliers. So again, that's things you would expect for fish that are really site attached. Jumping into another project that's using telemetry over much smaller spatial scales to look at lake whitefish habitat selection. The objectives of this project are essentially to map out spawning lake whitefish use of different spawning reefs, determine whether they're keying in on particular microhabitat features, and then trying to do some work comparing fish behavior and habitat selection between reefs to determine how consistent or generalized habitat selection is across different reef types. To do this, we're using something called positional telemetry. This is where you put a bunch of receivers all within tightly packed, usually 500 meters or less away from one another. They're all time synchronized, and then as acoustic tag fish swim through this matrix of receivers, anytime they're detected simultaneously on 3 or more receivers, you can use time difference of arrival techniques to accurately position them within the array. So this is just this figure is just a little example of a test array I put out with another project, and you can see the positioning is such as you can actually track individual fish movement through these arrays. We're doing this across multiple reefs. The data I'll share this morning is just from 2 of them, again from this Good Harbor Bay Reef, which is an offshore reef surrounded by deep water. It's heavily exposed to, or highly exposed to wind and wave action. That figure to the right there shows how we distributed acoustic receivers in 2022 on that reef to cover what we presumed would be the primary spawning areas. Then we also worked again at the Ingalls Point Reef on the west shore of Grand Traverse Bay. Again, the same figures showing where those acoustic receivers are located. This reef is much different. It's very protected. It's essentially kind of a big gravel or rock field that slides down from the shore. It's much smaller. So it has a lot of, other than substrate, it's very different in terms of its hydrodynamics. So let's jump in. So the questions we're mainly interested here is being able to map, seeing if we can make a correlation between fish habitat use on these, where they occur and the types of habitat they occur over, and then asking questions about whether habitat selection is similar or different across these different reef types. So jumping into some results for 2022, this period of time basically covers like November, December. This is where acoustic tagged lake whitefish were found on the reef, the Good Harbor Bay Reef in 2022. Thing that should jump out to you is that if you're out there, you would look over the boat edge and say, all of this area looks like great spotting habitat, but what you find is that the fish are very clustered and localized on particular habitat features. In this case, there's some kind of north-south running rocky ridges. We're still in the process. Other groups are still in the process of kind of characterizing what these particular physical features look like, but you can see the fish are all concentrated around these rock-like extensions out of the center, and there's very little use of this central shallow area, even though it, when you're there, it looks like great habitat. Same thing for Ingalls Point. Again, it's a fairly large spawning area, but the fish are all clustered in particular areas. Here, they're using a little bit larger proportion of the overall reef area. That large cluster there is kind of centered over a kind of a field of cobbles, gravel, and then there's another feature just to the west that's similar. It's a similar kind of rock ridge that we see in Good Harbor Bay that fish are also keying in on. What we're doing right now is we're taking these maps of fish habitat use, and we're gathering as much information as we can on various microhabitat layers that are collected at the same spatial scales as we've got fish data at. So we're relying heavily on Pete Esselman and those of you who are doing a lot of camera work to try to classify these bottom habitats in terms of substrate type, size, grain size, rugosity, zebra mussel coverage, etc. And we're taking these and combining them with the fish data to try to develop these resource selection models, which we can then use to apply and map out potential lake whitefish spawning areas in the other lake. This is just a couple of examples of using this information to estimate selectivity for depth and slope, which are really easy things you can just grab out of the digital elevation LiDAR models, which are easily available. And you can see there's not a ton of strong selection for depth, but the fish are more strongly selective for places with more intermediate slopes. So not flat bottom, but also not giant cliffs either. So something with them keying in on areas where there's some measurable slope to the bottom. So some takeaways here is that not all reef habitats created equal. We're definitely seeing these fish key in on specific parts or features of these reefs. It looks like slope and perhaps aspect may be already some important variables that are jumping out at us that are guiding habitat use of these fish. One of the things we're currently working on is looking at whether there's differences in habitat use and phenology amongst these reefs that might give us a clue to whether some of these populations are locally adapted. This figure to the right is basically the spawning phenology of the Good Harbor Bay and Ingalls Point fish, showing the number of unique IDs on those reefs over time. And pretty much the peaks overlap, but there is an indication that these Good Harbor Bay fish may be up on the reefs a little sooner than the Ingalls Point fish. So we'll do some more work with this to see if that's just a particular feature of this year or whether that occurs more regularly. And then even though we're still kind of in the early phases of pulling this all together, we've been really pleased that some of this data is already being used to guide some efforts. There's going to be some experimental mussel removals coming up on different reefs, and this information of how whitefish were using these habitats was used to help design some of the study plans for that work. So that's been really neat to see and cool, and hopefully this will be something that can help evaluate those particular success impact of those particular experiments as well. Just to wrap up, I want to give another plug for GLaDOS, which Chris Vandergoot introduced yesterday. A lot of this work wouldn't have been possible without the data sharing capabilities built into the GLaDOS network. So if you're interested in this project or others in Lake Michigan, please jump on their website. There's a lot of good information there. And with that, if there's any time for questions, I'd be happy to take those. So thank you very much.